I am your host and presenter for today. I am Subhashri Nag, and I'm a regional solution consultant for the Life Sciences and Scholarly Division here at Clarivate. And was a bench pharmacologist and a toxicologist prior to my present position. So let us talk about, though I have put it as industry key challenges, this is also applicable to academia. Okay, so we have participants today from different parts of the globe, right? And whatever whether you are from academia, whether you are from industry or a public research institution, drug discovery or research on the biomedical field is becoming more and more challenging by the day. There is a huge cost of doing wet lab experimentation, right? Now there is market share reduction in the sense that see, in the academic space, people will get funding if there is a certain commercial potential to the research that they are doing. And because there is a huge competition from generic and biosimilar compounds, there is decreased interest in funding very intensive and complicated, complex drug discovery projects. Now, all of these challenges mean that when you are starting with your drug discovery program, we need to devise a workflow where there is maximum return on investment on all the experimentation that you do. As we have already discussed, the cost of doing preclinical, clinical studies, subjects, the huge regulatory approvals that are needed, the cost of setting up the infrastructure that's very huge. The capex, as we say, the capital expenditure for biomedical sciences research is very high. So there needs to, if we can, streamline this workflow by getting solutions from trusted, reliable data that can actually accelerate innovation. And that is where solutions like Web of Science or Cortellis. So we will be discussing about two things today in the webinar. We will be discussing how a citation literature database like Web of Science can actually inform you about the discovery landscape or the research landscape, and that in turn can really help you streamline your research workflow. Then we'll also be talking about Cortellis, that is a specialized set of solutions that give you billions of hard data points. It's just not literature; they give you data points with regards to um, parameters being tested in experimentation about pharmacokinetic data points, but that is going to be just a small teaser. We will mostly so we intend to have this as a two-part webinar series. Today is part one in the series, and we will be talking more about the biomedical capabilities of the Web of Science database. We will be also introducing our attendees to Cortellis, but we will be talking more about Cortellis in the part two of this webinar series. Now, what is very, very unique about Clarivate is we have a very synergistic set of data sets. So when I talk about the various databases on the Web of Science platform that can give you information, trusted information about the biomedical research landscape, I'm talking about information from academic journals. I'm talking about, so I've just crossed our scientific records because it's not only scientific records, we also have from patent information, or we have data sets. We are getting information from publishers. We have chemical reactions. And the very unique thing, just starting to annotate. If you can see over here, this is something very unique. On Web of Science, you have access to more than 17 million full text papers. So for a lot of researchers, especially in academia, it is a big challenge for identifying the full text of the resources. That can be very hard. So when you are accessing a database like Web of Science, you also get unprecedented access to full text papers. And please note that these full text papers are just not from open access journals. Just give me a moment. They're just not from open access journals. Full text papers are from open access journals. They are from repositories. So even if a journal is behind a paywall, you might still get access to a full text paper through Web of Science. Stopping to annotate. So this becomes sort of your single source of truth. So what can you really do in order to have an informed and effective biomedical or pharma research and development strategy? You can map the global research landscape, track competition, explore the technology evolution, identify and engage with KOLs and principal investigators, assess competitive funding, and again, find low-level safety signals in a sea of noise. That's for pharmacovigilance. So what does the Web of Science core collection actually contain? The Web of Science core collection has more than 21,000 active peer-reviewed journals with conferences and books. And there's a very powerful bibliographic and cited reference network. And what it really helps you is citations connect ideas. So if when you are searching on a semantic database like PubMed or Google Scholar, these are the two mainstays of our biomedical researchers. One thing you must realize is you are entirely dependent on keywords. Now, sometimes, especially in a very rapidly evolving area, for example, coronavirus research, or the terminologies keep on shifting. Now, there are some papers on COVID-19, the early papers which came out from China, which do not use the terminology COVID-19. They are talking about a new virus from Wuhan, um, reports of a pneumonia-like disease. So they are important publications. They are the first publications that talk about how the disease evolved, what were the first symptoms that were seen. But if you just did a search for COVID-19, you would miss those papers. So these are the kind of papers that can be tagged or retrieved on Web of Science because of the citation connections across the platform. So today what we will also discuss is that Web of Science just does not contain the core collection. It contains around 35,000 journals across the platform. Now, why is that important? Because there are some very important subject specific. So again, starting to annotate. So there are some very important subject specific databases. So we will be talking about databases like Biological Abstract, Biosis Citation, and Biosis Previews. We will also be talking today a little bit about the patent collection, because the patent collection can be very helpful to the biomedical scientists for searching chemical structures. It allows you to do a structure search. By the way, the core collection, so the Science Citation Index also contains chemistry editions over here. You can see. So the chemistry editions actually also help you do structure searches. So you can actually do structure searches. Stopping to annotate. So using the Web of Science as a scientist, as a scientific information officer, what is the information that you can gather? You can find out what is the newest research coming out from academia, from industry. You can find out the collaborations, especially important for scientists to know where are the areas of alignment, who is doing what work, who are the key opinion leaders, what are they working on. We will be talking about each of these use cases one by one. And once I have shown you or run you through the deck, we'll actually go to the platform and see these examples live. So what I have actually done is, let us try to track a particular asset, in this case, a drug, and see how Web of Science can give you more information. Now, the drug that I have taken as an example is carfilzomib. Now, carfilzomib is a what is called a proteasome inhibitor. Now we know that cancer is an anti-cancer drug. Now we know that cancer is a reason of it occurs due to genomic instability, where what we can call the bad genes or the oncogenes are expressed more. They are upregulated as compared to the tumor suppressors. And in the cell, the cellular machinery, the proteasome acts as a kind of a garbage, collector or recycling of damaged proteins. So the proteasome can, in cancer, what happens is the proteasome is responsible because the oncogenes are the bad genes are upregulated. It causes increased degradation of the tumor suppressors by the proteasome. And what one approach has been, therefore, to identify molecules that can inhibit this proteasome machinery so as to increase the levels of these tumor suppressors and thereby prevent the classic markers of carcinogenesis, that is uncontrolled cell proliferation, angiogenesis, cell viability, etc. Now, going forward to this proteasome inhibitor carfilzomib, it's a very interesting story. So, in the early 1980s, Bristol Myers Squibb discovered a naturally Hello, am I audible? Yes, I am so sorry. I think I had lost for a moment. Okay, so just give me a moment to just make it full screen. Okay, so we were talking about following carfilzomib as an example of a assessed journey. So give me a moment. We had already talked about proteasome inhibitors. So this is very interesting. So I'll just start to annotate again. Starting to annotate again. So in the 1980s, Bristol Myers Squibb came up with this antibiotic. It is a naturally occurring antibiotic from a fungus, which is called epoxomycin. Now, very interestingly, if you can see my screen, there is a unusual epoxy ketone group over here. Now, to cut a small story short, this was taken up. Now, this drug was, it fell into disuse because people were not at the researchers at BMS were not sure about how, uh, you know, uh, um, what was the mechanism of action of the drug. So there was not much research done on it, but in the late 90s, researchers from Yale, they studied, so they found out about this drug using research sources like Web of Science. They were looking at, naturally reported antibiotics that had been shown to have anti-tumor activity, and they came up with this lead or analog candidate, and they found out that the way it works is by inhibiting the proteasome. And what they did was they created an analog, and that particular analog, which was called as YU101, so Yale University 101, and this particular analog was seen to be very effective. And by this time, in the early 2000s, another proteasome inhibitor, bortezomib, which is now marketed as Velcade, had already got approval to, as you know, as the first proteasome inhibitor. It was being used, and today, in fact, bortezomib is the standard of care for multiple myeloma. And so this gave them more, it gave them a boost. And with um, more refinement, what Cortellis was developed, which basically had a morpholino group that increases solubility, and it also had this part. Okay, so the morpholino group increases solubility. Morpholino group increases solubility, and this part is the important part, which is the pharmacophore. Okay, and eventually it was acquired by Amgen and marketed. So this is about a drug called carfilzomib. Now, if I wanted more information, so let us take the use case that I am a scientist who's working in an anti-cancer lab, and I would like to have more information about, Kyprolis, or I want to use the information available on proteasome inhibitors to quickly scan the landscape and understand what else can be done, what is the information associated with such drugs, right? Moving forward, so you can see carfilzomib with the morpholino group and the epoxy ketone. So it's Web of Science. Now, what is very interesting on Web of Science over here is, if you can see the screenshots, you can quickly find out who are the key opinion leaders. You can quickly do searches. Now, Web of Science allows you to, query with very complex queries. You can combine data sets together. These are things that you cannot do on PubMed or Google Scholar. So I'll just start annotating again. Now, if you are looking for drug information, okay, PubMed mostly peer-reviewed papers, right? But we know that for a lot of drugs, conference proceedings are very important. You do not get that on PubMed. Very rarely, until and unless the conference proceeding or meeting abstracts are part of a supplementary journal edition, you do not get it on PubMed necessarily. Secondly, on PubMed, you have a limited quantity of journals. If you looked at Web of Science, more than 21,000 journals, interdisciplinary. So let us say that there is a journal which is more on an organic synthesis, not covered by PubMed. We have a very rigorous criteria, but since we are multidisciplinary, if it's a good journal, if the editorial board has um, uh, mandated that the journal meets the quality criteria of Web of Science, it will probably there be on Web of Science. You might not find it on PubMed. Secondly, on PubMed, you cannot combine queries. Now, I can combine a query on Web of Science, for example. I want to look at only multiple myeloma drugs. And then I can do another data set for multiple myeloma and antibiotic and proteasome. So similar compounds like carfilzomib, which have a similar. So I know carfilzomib was developed from a naturally occurring antibiotic. So let us look. So I'm a biomedical scientist. I'm looking for ideas for, such drugs. Let us see any antibiotic is there if which does has been reported to have some kind of anti-tumor activity, or maybe an antibiotic which has been reported to have some kind of cell inhibitory property. I can then combine that with a data set talking about multiple myeloma, or for with some particular cell line that is used in multiple myeloma experiments. So I can get quickly to preclinical, to those publications that have published preclinical in vitro data on an antibiotic which has some activity in a human multiple myeloma cell line. Okay, so maybe I can do a HeLa. I can do a keyword search with HeLa. And now also, if you notice over here, and I'm still annotating, can you see there is a citation map associated with every publication? This can allow me to see all those citations. So for example, this particular one has 160 citations. I can look at these 160 citations and find out which are the components. So, amongst the citations, I can probably find compounds that have some anti-tumor activity and which have some natural or origin, or they are from they have some antibiotic property also, or they are of a microbial origin. So can you see how I can quickly scan the entire literature landscape using Web of Science? These kind of, going back and forth in searching and then collecting all the data sets together to give me one unique data set that I can quickly analyze is almost impossible to do on PubMed. So this is where the very, a very big unique proposition of Web of Science comes into play. I would also like to know which were the groups or the scientists involved in the study of anti-tumor antibiotics. I can quickly go to the analyze records. If you notice over here, these records, they can be analyzed. I can understand so I can see CM Cruz, right? I can also identify the funding grants. Okay, so for example, this grant, what are the papers associated with a particular grant? So another great use case example would be, I know a particular peer, I know that they got this particular grant. Okay, this is a peer, this is a competitor. I know that they have been awarded a particular grant and the grant number. That is very easy to find out from a lot of the public, sources that list the grant numbers awarded to the scientists. And then let us see, okay, how what are the production, what is the output of the grant? Using that grant, what are the kind of work that the particular public, competitor of mine is doing? I can even do an author search. Conversely, this also applies to the institutions themselves. If they want to know what has been the output and impact of particular grants that they have received from different funding agencies, this could be a great way to analyze that. We all know that when we submit grant proposals, one big factor that can actually be very helpful for ensuring that you get funded by that agency is a retrospective analysis of how successful you have been in your research using past grants or past funding from that agency, right? So that comes into your, as we say, letter of support. So this is where the research office can go to Web of Science. They can pick up, so okay, this particular professor, Professor Noor, I'm just taking a hypothetical example. The Professor Noor received this particular grant from this agency, and this grant has resulted in 5 publications in high impact journals. So that becomes a little support that this particular faculty has performed well in the past with funding from this agency, and therefore this agency can be persuaded to provide more funding in the future. So for all of you, somebody had a question that the flat slide is not moving. That is true. I'm just speaking right now. So we'll go to the next. By the way, Web of Science also has, a mobile interface. It is called My Research Assistant. Be sure to download that. So I'll take up a question that I have received in the chat window, and it's a very pertinent question before I move on. The poster has said that they have a subscription to a citation database, and as a university librarian, they want to understand the benefit of using Web of Science or this research scenario to our health science faculty. So what is the benefit of using Web of Science? First of all, you can identify breakthrough science very quickly. How do you identify breakthrough science? As I said, PubMed is not a citation database. When you're doing a search on PubMed, you do not get the citation analysis of a particular record. So you do not know how impactful that particular publication has been to the community in that particular area. On Web of Science, you can quickly sort by saying, I want to see in the area of breast cancer and research done with, let's say, this particular ZNY gene, the most impactful papers. I want to see publications only from a certain country. So there are a lot of filters that are available, analytical filters on Web of Science that can help you find the most impactful research, research from a certain affiliation, research from a certain institution, from a certain geography. One. Secondly, you can also go back into the citations. So for example, I am looking at a area of research, and I can go back to the cited references to see the evolution of a technology. How has a technology evolved? um, Another great way of use for your researchers to use Web of Sciences, let us say they have an idea in mind. Okay, so how they concretize that idea? Is it even worth investing in that idea? You can look at the publication trend. So I'm annotating. So some of the use cases would be, you can do a publication trend analysis. What about the number of publications in a particular field? Are they increasing or are they decreasing? Huge number of publications in a very short period of time, or an exponential increase in the number of publications, indicates an emerging area. Declining, You can also see which are the funding agencies associated with a particular, research area. Again, this can give your researchers ideas where to apply grants for. You can find out more information about a particular funding agency in terms of, for your own institution, which funding agencies have contributed to the research output and impact of your research institution. So that shows a positive relationship with the funding agencies. Your researchers can be encouraged to apply for more funding from these agencies because they have performed well in the past with them. You can identify potential collaborators. You're working in an area of, let's say, again, as I said, breast cancer and the P53 gene in your particular geography, in your country, which are the other researchers who have worked in that same field. If they have worked in the same field, how good are their publication in terms of citation impact? Where have they published? Are they publishing in high impact journals? You can find out the most impactful researchers in this way in a particular research area, in a particular geography, and they can be your potential collaborators. Finding out about funders, finding about collaborators, identifying novel technologies and breakthrough science. These are all the use cases that you can do with Web of Science. I hope I've answered this question. Um, can I have a confirmation if this is what you were looking for? We'll go forward. So as I said, in a particular area, you can find out who are the key opinion leaders. You can find out the most important papers. Can you see over here quickly? We have been able to, if you notice this, we have been able to filter them according to the time cited. Now, this is something very interesting. Web of Science data can be used in conjunction with scientific visualization software. In this case, I have done a co-occurrence analysis on VOS Viewer. Now, on Web of Science, one of the metadata points are author keywords and keywords plus. Keywords plus are specific terms which are added by Web of Science indexers. They are gleaned from the references cited by that publication. Okay, so they're tagged. So a paper might be talking about bortezomib, but they are not using the term proteasome inhibitor. But because this paper has cited a lot of proteasome inhibitor publications, we will tag that particular record with proteasome inhibitor too. So what I have done on VOS Viewer is I have uploaded the publications on proteasome inhibitors and I have chosen the top 1,000 publications in terms of citation impact in a particular time range. The time range is 2017 onwards. And then I have created a co-occurrence. So I have tried to see how are the different keywords appearing in that particular data set and how are they related to each other. So if you notice over here, uh, proteasome inhibitor, multiple myeloma, that shows that most of the publications in that data set belong to multiple myeloma, which makes sense, right? Can I just get a confirmation that my voice is audible, that I am audible? I think yes. So if you notice over here, obviously proteasome inhibitors, their major use in therapeutics is multiple myeloma. Okay. Now, very interestingly, when I look at carfilzomib, if you see, the warmer the color, more recent the papers are. So carfilzomib papers, they have all started coming after 2018 mostly in that data set. And there is a link between carfilzomib and cardiotoxicity. So even without reading any of the abstracts or analyzing the publications in detail, by doing this kind of a co-occurrence analysis, I can understand very easily that one of the toxicities associated with this particular drug is cardiotoxicity. Carfilzomib has some connection with immunotherapy, so it is probably used in combination with immunotherapy. And the drugs that are described in these publications are daratumumab, elotuzumab, which are monoclonal antibodies. So can you see how easily I can quickly have a bird's eye view as a researcher of a particular field? All I have done is exported the data set from Web of Science and uploaded into a software called VosViewer. This is a free software. I have put in the publication source of the software. You can also, if you all have Web of Science with you, please do check this out. It's a very wonderful way to visualize and make more sense of a scientific lab. There are several tools people have asked me. We are talking about VOS Viewer because of the reason being that it works very well with Web of Science. So in this case, I mean, we will not go into a comparative analysis of different softwares. There is BiblioShiny, there are R-based tools. Based on the user friendliness and uh, the alignment or the ease of use with WAS data. Okay, in some of the R-based tools, the data has to be massaged. The data has to be in a different format. This particular tool works very easily with the Web of Science data. There is no layer of data cleanup that is needed by the scientists themselves. It is also very user friendly. You just have to plug in the data to understand. So that's the reason we have chosen for this particular webinar the VOS viewer. But there are definitely different other scientific visualization tools also. Going forward. So some of the questions, for example, we have a very interesting field on Web of Science called the hot paper. Now, in a particular field, if you notice a field like proteasome, and if you notice a field like breast cancer, these are very saturated fields. A lot of people are doing work on that. And we all know that citations are a function of time. So sometimes when you're looking at the citation data, you might come across papers which are 5 years old, which are not really relevant to the very content, especially in biomedical research, where the pace of research is extremely high. It might not make sense. So in that case, hot papers can be very, very useful. You can understand key opinion leaders. Just to reiterate, hot papers are those papers that have been published in the last 2 years, and they are trending in the sense that they have got most of the citations in the last 2 months. You can quickly identify experts. You can have more information about an expert's profile. For example, Mohammed Moti, who has a lot of publications on oncoimmunology and is from France. You can find out very quickly what is the kind of information that they have done. Uh, Going forward, backward once to answer a question. What are hot papers? Hot papers are papers that have been published in the past 2 years. Okay, so they reflect contemporary research interests, and they have accrued the most citations in the past 2 months. So the number of citations they have accrued have made them go to the 0.1% of the recently cited papers in their field. And this data can be used to understand where the leading research in your area is being produced and who is funding it. You can find out which are the corporations universities are working with. So for example, in this particular example, we have taken immuno-oncology, and we have seen that Johns Hopkins works with National Institutes of Health, and these are different universities. Now, you can understand and find this out to gauge the opportunity areas. Maybe if you can even do a comparative analysis. I will now go to the subject specific databases. So we have the chemistry database, this is really important. This is really interesting. Many of you probably do not know, but Web of Science actually allows you to do structure searches also. Now, in this case, I have drawn the structure of rosmarinic acid. It's a natural polyphenolic compound which is found in rosemary, the herb. And you can try to see, you can do a substructure search, and it gives you a lot of information about the chemical reaction. So this is a reaction detail for the synthesis of this particular compound, and you can see all the reaction conditions are provided along with this, but data. This can be very, very helpful for a lot of biomedical scientists are also working in the area of medicinal chemistry and organic synthesis. So especially for drugs, if you want to know which are the patented chemistry routes or how have certain drugs be synthesized, this can be very, very helpful. And this is one integrated platform. And so, for example, in carfilzomib, this is from an Indian patent. Okay, so all the reaction steps, if you notice over here, are given, and each of these reaction steps actually has detailed commentary on reactants, catalysts, and conditions, just like this. We also have the Derwent Innovation Index on Web of Science, and it allows you to get information for all patented compounds. So for example, over here, I'm searching for a beta carbolane nucleus, and it tells me which are the different drugs which have this particular nucleus within it. And the best part about it, it is great for understanding patent. The Durbin Innovation Index, this is all the patent data. If you remember, earlier chemistry index had information from both patent as well as scientific literature. DII, or Derwent Innovation Index, gives you information from 90 million patents, and they have a descriptive title and abstract, and also has citation data. Very importantly, not only do you get information about the patent, you can also find out what scientific literature they have cited and what, so you can really find out how has the technology evolved. Next, we go to the specialized index databases like Biosis, which are there on Web of Science. Now, Biosis Citation Index um, is a biology specific database, and what is its purview or its coverage is of course more specialized than that of Web of Science. There are more than 5,300 journals, but it reveals more accurate and context sensitive results because there are specific controlled vocabulary thesaurus associated with this particular database. So for example, I can look at carfilzomib and look into the concept of toxicology to find all the safety and adverse effects associated with that particular drug. This would be very helpful to both health sciences, even doctors and clinicians. Today, I know our attendees have a lot of medical professionals, especially if you're a busy doctor, you want to do a literature search. Web of Science is much more richer than PubMed in letting you get exactly, so you can do toxicology, carfilzomib, and it also has a hierarchical. So in toxicology, you can go to teratology, fetal toxicity. With this, I conclude the major portion that we wanted to talk about the Web of Science database. Now, having seen or had a flavor of this database, I'm sure one question that you all are very keen to know is that what about if I really needed some data points? For example, I am a pharmacologist. I would really I'd love to know what is the dose that somebody used. Now you can find that data information on Web of Science, but you'd have to read the abstract. You'll have to deep dive into that particular publication. But what if you had a data set which could give you, okay, which genes are interacting with each other in a particular disease state, or if A interacts with B, what happens in the body? Or let's say there is a drug, carfilzomib. What are the experimental models associated with carfilzomib? What are the PK parameters of this particular drug? Um, what are the doses that are used in children? Are there any drug-drug interactions that I, as a clinician, should be, aware of? These kind of hard data points are not available on a literature database, but we do have discovery and preclinical data at Clarivate too. We have Cortellis drug discovery. We have discovery data in terms of Metacore. We also have a toxicology database called OFFIX. Now, today I'll just introduce the databases to you. We will be covering all this discovery and preclinical data in the part 2 of this webinar series. So as I said, what if we had a database that could quickly give me an idea about human pathways with containing target genes that can help me develop better hypothesis as to whether this is the right target for a particular disease? Should I be working on that target? Or if I could even upload my own gene array data and try to see the make sense of the differentially regulated genes and fit them into some pathway in context of a disease. That is where Metacore comes. Metacore is basically a pathway analysis tool that is provided by Clarivate, and that helps you see molecular interactions in biological systems and their signaling pathways in humans. We record more than 2.6 million molecular interactions, and along with that, we also give you the directionality. So A is affecting B or B is affecting A. If that there is an interaction, what kind of interaction is it? Is it a post-translational modification? Is it a covalent binding? Is it an indirect modulation of the gene? How does that interaction happen? Next is what we are going to talk about. If I am a clinician or a pharmacologist, I would like to have integrated biology, chemistry, and pharmacology data at one place. Um, I would like to benchmark my compound. So I am working on some compound in my lab. I would like to see how, and I have my own data with regards to, let's say, toxicity, with regards to the enzyme inhibition, etc. How is it comparing? How does that data compare to the best in class? You can identify and prioritize genes and targets. So all this information is there on one integrated database called the Cortellis Drug Discovery Intelligence. And how that can help you is, let's say your institution's focus is to identify and validate targets and understand their pathways. We have Metacore, which leverages high quality granular systems biology candidate. You can identify assays to test your lead candidates by seeing data from CDDI. You can keep an eye on the constantly evolving competitive landscape. So we have a lot of pipeline data also. So with this is a very small introduction to CDDI. I will show some examples on the live platform too, but we will be covering all of that in more detail in the part 2 of the webinar series. So with this, I will stop sharing, and the webinar is not over. We will actually be going to the live platforms to do some searches over there. We still have 10 minutes, so please let your questions come in. And I will be sharing now my browser window. So can I please have a confirmation that people can see my browser window? Okay, perfect. So this is how the Web of Science platform looks like. Now, if you remember right in the beginning, we started by talking about how there's a lot of full text information on Web of Science. Now, every publication on Web of Science is categorized into a certain subject area. There are more than 250 such subject areas on Web of Science. Now, I have just put BioStar, so I'm not covering the clinical medicine parts, right? But I still am covering biochemistry and I'm covering biology and biophysics, etc. And if I do a search on Web of Science, I can see there are 7.7 million records. And what is the best part of it? Around 30% of this data you can access freely. If I can just annotate my screen once again. Can people, can you see where we have 2.1 million free records. And this is just a small, part of the iceberg. I have not taken any clinical medicine. So we have neurosciences, we have psychiatry, we have, internal medicine. So let's look at internal medicine just as an example. Stopping to annotate. So let's go over here. And I'm just looking at medicine. So just internal medicine. Internal medicine itself has 2.8 million papers. And out of that, 700,000, almost 700,000. And I can actually also put, let us see how much information we have in the past. See, I can quickly look. Refine. 356,000 papers published in the last 4 years. And out of that, oh wow, this is amazing. Can you see out of 356,000 papers, 234,000 papers you can find full text. So the basic idea of showing all of this is that the coverage on Web of Science with regards to biomedical literature is unprecedented, much more easier to find, quicker to analyze than PubMed. Now, let's look by the most highly cited papers. Okay, some of the highly cited papers are all related to COVID-19 in Wuhan. Now, I can also quickly do an analysis like this. Let's do another search. Topic. I'm just choosing COVID-19. That's it. Okay, huge number of papers on COVID-19. Now, what I would like to do now over here is let's go back to our advanced search. So if you remember our internal medicine papers, we had a lot of papers on COVID-19, right? I don't want these papers. So what I can do is add to query. This one, add with a not. So I want 5, but not 6. So now I come with 331,000 publications. So can you see over here? This is really interesting. I am still getting some of the papers on this. Is the reason why, if you remember, we were talking about citations and how terminologies change. Though I took away the keyword COVID-19, can you see this Lancet paper from February 2020 that talks about the 2019 novel coronavirus but does not use the term COVID-19? Yes, all the publications over here can be exported to EndNote Online. Okay, so or even to Mendeley, you can use BibTeX. You can also use RIS to for other reference software. So there are different ways. I can just click over here, whichever papers I would like to, export. So again, using a citation database like Web of Science allows you to export this information directly to a reference manager, and a reference manager like EndNote on the online version is freely available. So somebody has asked the question. Yes, early papers are probably best searched with Wuhan coronavirus, Wuhan pneumonia. That's correct. You can quickly sift and see the review articles. You can see the open access. Now, if you notice over here, I do have a EndNote plugin. This is called EndNote Click. What it does is that it quickly identifies the free full text. So if I just click on this paper, it will work in the background and identify the free full text, and it will save it in a it will save this in a particular in my virtual locker. Can you see view PDF? And this open access version has been saved in my locker. Another thing that Web of Science can be very helpful is understanding the quality of a particular journal. Okay, so there are some journals on Web of Science which have not yet received, and yes, you can see the abstracts also on the dataset page, though I don't really recommend. It just makes it a little bit, difficult to analyze. The best way to read the abstracts quickly would be download or export that entire dataset as an Excel file. Okay, so it will be you will have all the abstracts in one column and quickly read them on the Excel file. Having said that, let us say as a medical researcher now, all over the globe, one of the biggest problems that have plagued the biomedical research community is the rise of the so-called predatory journals, which has been disproportionately high in the biomedical space. And unfortunately, a lot of these journals emerge from countries like India, some from Southeast Asia, but a lot from India, some from Iran, so these geographies have been shown to contribute to these predatory journals. Now, how, and there are very strict rules. For example, in India, you have the Medical Council of India, the UGC, who have strict rules that where can people publish. Now, am trying to look at a new journal. How do I know that the journal is good enough? Is it a journal that I should be publishing? Now, of course, the first thing that you should look for is whether that journal is a part of Web of Science or not. That itself tells you that it has undergone the minimum, editorial evaluation to ensure it is not predatory. So let us take, for example, a journal called Curious. Search 18,000 titles from Curious. I can quickly look over at the publication years. So 2021, 6,000, so a lot of papers. Now I want to look at what is happening in these 3 years, because 2021 citations are still coming in. We are just in March 2022. So in order to get a good idea of the citation impact of the papers in a particular data set, I have taken a year previous, so 2018, 19, and 2020. Let us go to the citation report and try to understand what is the average citation, where are the citations coming from, the citation footprint. All this information can be very helpful. So again, for the university librarians who are attending this session, if you'd like to advise your researchers whether to go for a particular journal for publishing or not, this could be a great way to do that. So I can see this is the average per item, 2.15, which is low because this is an internal general medicine paper journal. And I can even analyze. And one good thing is that if you notice over here, the self citation is quite low. Okay, now we do not want to publish in a journal that has high rates of self citations. I can analyze. So now I am not analyzing the publications in that particular journal. I am analyzing the publications which are citing the articles in that particular journal, which is curious. And let us see where are the citations coming from. Which other journals are citing. Okay, Curious itself has cited, but I have citations from BMJ case reports, World Neurosurgery, some from scientific reports, plus one. Okay, now let us look at where are the citations coming from as in institutions. Okay, so I have from UC System, Harvard, Mayo Clinic. That's a good thing because that means people from some of the top medical colleges and drug research institutions are actually citing papers of in this journal. So they're reading Curious. So some of the key opinion leaders in the field are reading. So this can be a great way to orient your researchers. Where should you be publishing? Quickly understand whether a journal is worth publishing in or not. Finally, going to CDDI. If you remember, Cortellis Drug Discovery Intelligence, I can quickly type in the name of a drug. In this case, I'm going to be typing in carfilzomib, and I can have a very good idea of what is happening in that particular drug space. So there are 7 drugs. There are 717 experimental pharmacology records associated with this drug. I can click over there. Can you see? It tells me what pharmacological activity, what is the material, the assay. Amazing data for the pharmacologists or any discovery scientist to plan and execute research. This would be very, very helpful for really decreasing that, level of uncertainty in the research discovery workflow. With this, I leave you with enough information to how to streamline your research and discovery workflow. We are slightly over the hour. I'm stopping to record, but I can still take questions.